All right, you guys, can you hear me?
Yeah.
Yeah.
Okay, cool. All right. How was your weekend?
Well, I'm guessing we all were doing our senior thesis presentations.
Wasn't everybody working on senior thesis stuff this weekend, myself included? How's that going?
Just ready to be done with it.
Okay.
How long are they supposed to be again?
I don't recall.
15 minutes, 5 minutes for questions.
15.5. Okay. Yeah, that's about normal. Yeah. So I was doing an analysis for Chris, and it's always a bad sign when you get to Monday morning and this thing is open at the bottom of your screen. Are you guys familiar with Terminal? Yeah, this is where you go where you're basically doing line coding on the hard drive of your computer, which means that you have a great potential to totally screw up your computer while you're in the inner guts. It's buried away in utilities, which is where you can't find it very easily because computer manufacturers don't want you mucking around on the inside of your computer like that. So that's always a bad sign when you boot up your computer on Monday morning and the terminal is still open from your weekend activities. So anyway, it's almost over for you guys in terms of senior thesis, at least in terms of the presentation. You guys still have to write a report if you haven't started that yet, but
I've not written a word.
No, not written a word. Well, at least you have a proposal laying around somewhere that has some resemblance to your final project.
We'll be copying, pasting most of that probably.
That is, I think, perfectly allowed, although you might want to check with your research advisor just to make sure. But quite frankly, that's one of the purposes of us having you do that back during your junior year, so that when you do get to this point, you're not starting from scratch, and that's always nice. In the same way, for your presentation, you've also given, you know, in other venues, you've given kind of an introduction to what your project is about. You can also recycle a bunch of that. So it may not seem like it, but we have made you do things in the past that hopefully makes this past weekend and today a little less stressful, although probably still stressful enough. So anyway, that is that. I will try and get through the remaining material on interspecific interactions as efficiently as I can, so that I can let you guys go and stress about things that you really want to be stressing about. So we were at the end of talking about The vomeronasal organ, also known sometimes as the Jacobson's organ, although that's old terminology. People now pretty much just
You
refer
can't
to
see
it
the
as.
slides.
I know I'm getting there.
Okay, just making sure.
Yeah, I'm rearranging things on my screens, and I was just about to grab the share screen button.
You're good. Sometimes it's just hard to tell when you start talking about things if you want us to see the slides or not.
Yeah, sometimes I'm talking to fill in space while I'm doing things, and this happened to be one of those cases, but thank you for taking me to task, Kelsey. All right, so we were talking about trailing behavior, and can you see the slides now? Yeah.
Yeah.
So we were talking about the vomeronasal organ. It used to be called, well, it still is called the Jacobson's organ, but we've kind of moved away from that terminology. We talked about prey trailing behavior for after a rattlesnake, for example, strikes a mammal, and you can lay the mammal down on a sheet of paper, and the snake will follow the scent markers of that mammal, and it does better if it's actually struck the mammal than if it hasn't struck the animal. And so this is kind of an old experiment from the 80s that looked at the ability of snakes to do this with their forked tongues and vomeronasal organ. So we were about to move on to foraging modes. And these foraging modes exist on a continuum, but we oftentimes classify organisms as either sit and wait predators or active foragers, active searchers. And sit and wait predators do exactly what you think of them as doing. They sit in one spot and they wait for prey to come by. And when the prey come by, they grab it, and that's how they find their prey. And it isn't so much them finding their prey as much as their prey kind of stumbling into them. Active searchers, on the other hand, are constantly moving around the environment looking for prey. And so these two ends of this continuum have certain characteristics that are correlated with these two particular lifestyles. So for example, sit and wait predators will oftentimes rely on visual cues, whereas active searchers are oftentimes relying on vomerofaction, using a vomeronasal organ. Now that you know that that's two types of differences, sit and wait predators are waiting for animals to kind of walk past their field of vision, and once they see them, they will attack them. Whereas active searchers are actually sniffing out their prey in different places. What would you expect their tongue to be like, knowing these two things about them? Knowing that active searchers are going to be using vomerofaction.
Would active be forked?
What's that?
Would active be forked?
Yes. So sit and wait foragers are going to have fleshy tongues that they just like eject out of the mouth, stick to the prey going by and pulling that thing in, as opposed to active searchers are going to have these forked tongues that you oftentimes find associated with the vomeronasal organ. From the standpoint of sit and wait predators, they're going to obviously be finding more obvious prey, as opposed to active searchers that are going to be finding hidden prey because they're finding them by other cues other than seeing them. What are some other differences that we might expect to see in terms of differences in foraging mode that are correlated characters? I think of a horned lizard as the classic sit and wait predator, and I think of a T-ed lizard, something like Aspidicilla sexlineatus, as an active foraging species. What do you remember about those two body shapes?
The sit and wait should have like a stockier body. Like it shouldn't be as slender.
It is sit and wait predator is not going to be a sprinter. It's going to be short, stocky, short legs. And what do you notice about Aspidicilla sexlineatus?
Doesn't it have a long tail?
It does have a long tail. It also has a long
Body?
A long body, yeah. So sit and wait predators have stout bodies, short tails, and short legs. And active searchers have longer, slender bodies. Both the body's legs and tail, all those things are going to be longer and more slender as well. Usually you find sit and wait predators with low metabolic rates, and active searchers will generally have higher metabolic rates. And I think those are the main things. So with this particular way of finding food, you get all these other things that tend to be found in, if you're an active forager, you tend to have these types of characters. If you're a sit and wait predator, you tend to have these other types of characteristics. And while that's generally true, there are always exceptions, but this is a general kind of finding. Foraging mode can be phylogenetically constrained. So for example, when you think of teid lizards, think of Aspidicilus, all of the race runners and the tegus and things like this that are in that family, they are all active foragers for example. Varanids, almost all of those would be classified as active foragers. Skinks, I realize that the only time that you have really observed skinks is when they're curled up under a rock trying to get away from the cold. But when it's not so cold out, they're actually moving actively through the leaf litter in the forest that they occur in here in the United States. When I was a graduate student working in East Texas, you could go out to any of those forests, and as you walk through the leaf litter, you would literally see ground skinks, Ginsella lateralis, just scurrying away from you in the leaf litter. They are diurnal, they are active, they're constantly out there darting about foraging. Lacertidae, which is the old world equivalent of the iguanidae, are also active foragers. And then when you look at the family Iguanidae, you find that those are primarily sit and wait predators, with the exception of herbivores. Why are herbivores not sit and wait predators?
Because plants don't run away.
Because plants don't ever run across your visual field of search. And so obviously, even within a particular family, you're going to find that there's variation, but there are these broad scale sorts of things. And all of these examples are lizards, but you could do the same thing for snakes. So within the snakes that we've looked at, Colubridae and, things in the family Colubridae, like Coluber constrictor and Pantherophis obsoletus, those are actively foraging species. They tend to be longer, they tend to be thinner. When you look at the pit vipers, things like the copperhead and the cottonmouth, those are shorter and stockier. Those are generally sit and wait predators as opposed to active foragers. So it applies to any group of organisms you could classify in this way. This was a classification system that was first set up with lizards in mind, but the fact of the matter is you could use this for any group of organisms in reality. And it has been applied to any group of organisms. So second to the last set of interactions is parasitism. So we talked about this a little bit last time. Parasitism and predation can both be classified as interactions where one species benefits at the expense of the other species. So it's a net positive for one species and a net negative for the other species. So how does parasitism differ from predation from the standpoint of the size of the parasite and host versus the size of the predator and prey?
The parasite. Oh, sorry, Elizabeth, go for it.
Okay, the size of the parasite should be a lot smaller than the host, whereas the predator should be around the same size as the prey.
Or bigger. Yep. What about from the standpoint of the length of the time that the interaction occurs?
The parasitic relationship would be like longer term.
Yep. Why is that?
because the parasite's relying on the host for like all its nutrients, or nutrients or something, versus like a predator just like attacking prey and eating and then being done.
Yeah, I mean, with a predator-prey interaction, you attack and kill the thing, and then that's it from the standpoint of the prey. It's over. And so this actually then folds into the last one. What are the final consequences to the species that's negatively affected by the interaction? In the case of a predator-prey interaction, the prey. In the case of parasites, the host.
The prey is dead or severely injured, and then the host is just negatively affected. I guess maybe dead sometimes after a certain point.
Yeah, but it's in the best interest of the parasite to not kill its host. So generally, the final consequence of predator-prey interaction is going to be death, and the final consequence of parasitism is you're still alive. You may not have the same quality of life that you had before you were parasitized, but you're still around and kicking. And it is possible that you could succumb to the long-term effects of parasitism, but you live for a long period of time because the parasite itself is relying upon you for shelter, food, everything that it needs is in the host. So in these fundamental ways, parasitism differs from predation, even though they would generally be classified in broad terms as positive for the predator or the parasite and negative for the prey or the host. So we can think of parasitism in amphibians and reptiles by dividing it up into internal parasites versus external parasites. But even within those broad classifications, you might think of the different kinds of organisms that afflict amphibians and reptiles. Your book talks briefly about lizard malaria. This was something that was discovered by a guy named Joseph Schall. He found in anoles in particular that live in tropical regions. They get fed upon by mosquitoes, and then the mosquitoes transfer the protozoans that cause malaria in humans. They will also transfer those parasites to lizards. And while it doesn't have the same systemic effects, it does reduce their fitness somewhat when they do come into contact with these protozoa and parasites. When we think of multicellular organisms, we oftentimes think of nematode worms. Nematodes are surprisingly common out there in nature, and there are a number of them that are parasitic. The book mentions two case studies, one looking at cane toads that are found in Australia. So the cane toad, which is in the genus Rhinella, it's undergone a genus change. I think its correct name now is Rhinella marina. Used to be Bufo marinus. It's one of the largest toads in the world. It was introduced into Australia quite some time ago and has basically run rampant. Because my library is a constant source of fascination, there is even a short book called Cane Toads, An Unnatural History. And there's even a documentary entitled Cane Toads, An Unnatural History. I believe that grew out of this book. It's a small book, it's easy reading. You should read it to your children if you grow up and have children. So this thing is spread throughout Eastern Australia and has gotten up into Northern Australia. And as it's gotten into Northern Australia, the rate of spread of this animal has really increased. And what has happened is when it was first introduced, those species were afflicted by nematode worm parasites that affected their fitness enough that they didn't reproduce as well. And so their rate of expansion of their geographic range in Australia was actually relatively slow compared to once they got into the northern part of Australia, and now they're moving westward along the northern coast of Australia. They have reached an area where these nematode parasites are not as common, and it has actually affected the rate of spread such that when they're not inhibited by these nematode worms, they're actually spreading much faster than they used to. In the case of horned lizards, there's a nematode worm that affects the ant species that they feed upon. So when the nematode worms come out of the horned lizard, the female basically lays all of its eggs. Those eggs are encysted. And when the ants find those eggs, they mistake them for seeds. These are seed harvesting ants. They bring those seeds into their nest. They hatch out. The nematodes go into the larvae of the ants. They end up getting into the adult ant. When the horned lizard eats the adult ant, then the nematode worm gets into the horned lizard, gets into the gut of the horned lizard, and completes its life cycle. So in all these cases, the amphibians and reptiles are basically serving as kind of secondary hosts for these animals. There are flukes and a type of annelid worm that can once again be found in water. These things will actually infect tadpoles. And then as those tadpoles are going through metamorphosis, the flukes insist themselves down where the limb buds are coming off, are developing. And so larval development is disrupted because there are these flukes that have formed cysts around the limb buds. And in some cases, it affects the frog by it loses limbs, or in some cases can affect the frog. In this case, where you have a frog with two normal hind limbs, and then there are these two aberrant hind limbs coming out. And these are all due to changes induced by these flukes that are hanging out, going through their dormant stages down where the limb buds are developing, and then causing these limb deformations. We also have external parasites. It's really common to see leeches on the outside of turtles. Any turtle that you pick up anywhere throughout the southeastern United States, I would be very surprised if you didn't find either leeches or scarring from past leech infestations. But among the squamates, a common thing that you have are ticks and mites. And the thing about ticks, and especially mites, is they tend to only occur in certain places. And they tend to occur on these animals where there are little folds of skin. And so you have nuchal folds, axillary folds, post-axillary folds, inguinal folds, and postfemoral folds. And you tend to find these mites and ticks hanging out in those areas. And one of the things that your book talks about is that these folds may actually have evolved as places for mites to go, so that mites will hang out there rather than going into the ear of the animal or hanging out around the eye of the animal, although you can see that there's some eyes around this Texas horned lizard here. What's weird about this is different species are differently susceptible to mites. So for example, I work in Galveston, and in Galveston there are two species of introduced geckos. Hemidactylus tursicus is what Zach and Chris have been working on. And then we have Certipodium scabrum, which is a more recent introduction. And even though these two lizards are taken out of the same habitat, in some cases taken off of the same wall, in some cases collected just inches from one another, Certipodium scabrum almost always has mites, either around the eye or back here in the axillary or the inguinal body folds. And Mediterranean geckos never have mites on them. And so two species in a place where these mites clearly occur, one of them is always infested with them and the other one is not. And so there's quite a lot of variation in terms of one's tendency to be afflicted by mites. So this brings us finally to predator-prey interactions. And amphibians and reptiles end up being the prey for lots of different organisms, primarily because they are small. And they are small, so there are lots of potential predators that are larger than them, and so they're at something of a disadvantage. But the type of predator that you are preyed upon varies somewhat depending upon what life stage you are. And so when you think about amphibians in particular, amphibians start off as eggs, which are very susceptible to predation because they don't have any protection at all, with a few exceptions. And then you have the larval forms that are a little bit better to protect themselves because they can just swim away from any threat, but they live in water, so there are a different suite of predators that potentially feed on larvae than feed on the adults once they're out on land. So here we have a couple of examples of wasps that are actually feeding on frog eggs with a developing larva inside that are adhered to this leaf, and they will actually pick up, in some cases, the egg and fly it back to their nest. Here we have, once again, a cat-eyed snake in the genus Leptodyra, feeding on eggs that are once again glued to the surface of this leaf in one of the tropical species that lay their eggs on leaves. There's a number of them that do this. These are a different suite of predators than, for example, the predators that feed on the adults. So the left-hand frame here, this is a bat that is feeding on a frog called the Tungara frog. It's the frog Physolemus pustulosus. This is a frog that occurs in the New World tropics. That is the preferred food of one particular type of frog-eating bat. Here's a kookaburra eating a limbless skink, and here's a giant water bird, once again killing and eating another Physolemus pustulosus. So amphibians in particular are unique among vertebrates in that they are oftentimes small enough that they become prey for invertebrates. In addition to eggs being consumed by a wasp, you also see larvae being consumed by water bugs, crayfish, dragonfly, damselfly larvae, etc., while they're in the larval stages. But even when the frogs metamorphose and they're in their little froglet stage, there are plenty of invertebrate predators that are capable of feeding on them. And so this leads to what's sometimes referred to in the literature as feeding loops. Normally, we think of, plants being eaten by herbivores, which are eaten by primary predators, which are then eaten by secondary predators, which are then eaten by tertiary predators. And we generally don't get much higher than tertiary predators because we just kind of run out of energy, because we lose roughly 10% of the energy as you move from plants to herbivores. Another 10% as you move from herbivores to primary predators. But in some cases, you have animals that don't follow this linear sort of pattern. You get a situation where this predator can feed on this predator, but this predator can also feed on this predator. And that's the case oftentimes with amphibians and their insect predators. When this frog is really small, it is small enough to be eaten by this insect. But as this frog grows up, and if it avoids predation when it's younger, it will get to the point where it is then large enough to feed on this insect. And so we oftentimes see amphibians in particular involved in these feeding loops. We can also see reptiles involved in this also. This happens in Puerto Rico, where I used to work. There is a thing called an Amble Pigeon. I don't know if you guys are familiar with Amble Pigeons. I should have pulled this up yesterday when I was thinking about this and didn't. Can you guys still see my screen? I'm on Google.
Yeah, we can see it.
Amble pigeons are interesting organisms. They look like this. Wait, whoa, go back. Wow, that looks strangely like a photo I took. wonder if somebody stole that photo. Anyway, these guys are crazy looking. They have these large chelicerae. They have spikes on them. That's what some of the longer limbed ones look like. And they're big enough that they're bigger than they're bigger than even the largest frogs in Puerto Rico, the largest coquis in Puerto Rico. And they will be found feeding on frogs. They'll be found feeding on lizards that they find sleeping on leaves at night. And so when they're adults, they are quite large and can eat these other vertebrates. But when they're babies, they're quite small and they will be fed upon by lizards and frogs and other things. And so this is an example of a feeding loop in Puerto Rico where Amble pigeons as adults can feed on other vertebrates, but other vertebrates can feed on Amble pigeons when they are small enough. Yeah, they're scary. They're frightening when you see them at night the first time. Let's go back. Let's go back to this. Back to who preys on amphibians and reptiles? Yeah, okay. So your book, and I had never taught this far into the book yet. So I was gratified to see that when your book talks about predation, it essentially follows the predation sequence. So if you notice in the section on predation, it basically is going through all of the ways in which amphibians and reptiles avoid being found, all the ways in which they avoid being recognized, all the ways in which they avoid being caught and killed. And it's interesting to me that essentially this portion of the book chapter is essentially arranged the way the predation sequence is done, which is nice because it shows me that my adherence to the predation sequence is not just me being unique, it's a general feature that people recognize. So your book talks at some length about horned lizards and especially about the blood squirting behavior of horned lizards. But I thought it would be useful to think about the horned lizard from a number of different perspectives. The first avenue of defense from predation is actually to just never come into contact with a predator by not being found. So during the search phase, the predator is looking for prey. And of course, what does the horned lizard have at its advantage in this situation?
Camouflage.
What's that?
It like blends into its environment.
Yeah, so it's got camouflage or it is experiencing or exhibiting crypsis. And so you see this lizard that blends in very well with its background. If you can avoid being found or avoid being recognized as prey and you just look like a set of pebbles laying there on the ground, you've basically stopped the predation sequence in these early stages so that you don't move from there on to being captured. Once it has been noticed and recognized as prey, one of the ways that it can avoid moving to the catching of prey stage, what is this lizard doing in this frame down here in the lower left? This is a roadrunner going to attack this little horned lizard. You'll see horned lizards do this if you ever find one and pick one up.
It looks like it's trying to make it look bigger.
Yeah, it's puffing itself up full of air, kind of like toads do, to make itself so big that the roadrunner either can't get its beak around it or can get its beak around it but can't get it down its throat. And so it can essentially make itself larger to avoid being captured. The other thing that it can do is it actually has chemical defenses. They squirt blood out of their eyes, and this is especially triggered by the scent of dog saliva. This has actually been studied in some detail. And evidently the blood is very, very distasteful. So here's a bobcat that has been exposed to horned lizard blood, and it's basically gagging and about to throw up. So once it has been captured, it avoids moving from the catching prey stage of the predation sequence to being killed and eaten, once again through chemical defenses, like squirting blood out of the eyes. What is the other defense that horned lizards have to help them avoid, after being captured, becoming food, being killed and eaten? You guys were exposed to this a little at the very beginning of the evolution and ecology. What kind of lizard is this?
Texas horned lizard.
It's a horned lizard, and so it also has not only chemical defenses, it also has
Elongated horns.
yeah, it has spines and elongated horns, which once again can prevent it from being killed. You guys were exposed to a paper back in evolution and ecology where they looked at the horn length of lizards that had been killed by shrikes versus ones that had not been killed by shrikes, and found that those that had been killed by shrikes had shorter horn lengths than the ones that survived. And so horned lizards essentially represent for prey a variety of different ways in which you can avoid moving from one phase of the predation sequence to the next phase of the predation sequence, through crypsis, through avoiding being captured by making yourself larger in this case, weapons of defense, the horns, chemical weaponry like squirting blood out of the eyes. All of these things essentially keep you out of the gut of the predator. Your book talks about postmatism and mimicry a lot, because we see a lot of postmatism in amphibians and reptiles. Amphibians in particular, because amphibians create a lot of skin toxins in the glands of their skin. Here we have some dendrobatid toads. Here we have one of the harlequin frogs from Central and South America. Here we have Bombino orientalis, the fire-bellied toads from Europe and Asia. And here we have a fire salamander from Europe. This is some work done back at UT Arlington when I was a student there by Butch Brody. He actually found that fire salamanders, they have muscles that go around their toxin glands, such that they can direct their toxin gland to squirt the toxin at the face of the predator. So here you can see Butch Brody is prodding this salamander, and the salamander is squirting this toxin in the direction of where the pokey stick is coming from. So these guys produce all sorts of toxins, and the toxins themselves can come from all sorts of different places. In some cases, frogs and toads will eat ants, and they take the substances that are in the ants and they metabolize them into toxins. In some cases, it appears that the animals themselves are creating the toxin endogenously. They generate it themselves. In the case of some snakes will eat toads. The toads themselves have the toxins, and the snakes will actually sequester the toxin. And so the group of frogs, in this case the mantelids and a bunch of the dendrobatids, they get toxins from the food that they eat. Thamnophis can get toxins from the salamanders that they eat, even though the salamanders themselves are generating it independently of their diets. And then Rhabdophis get bufotoxins, and also it's thought that heterodon. So when we talked about the hognose snakes, I mentioned that they are toad specialists, and your field guide also mentioned the fact that they play dead as a defensive mechanism. One of the reasons that some herpetologists think that that hognose snakes feign death is that they've actually sequestered bufotoxins from their food, and when they're put under stress, they start metabolizing fat, which is where presumably these bufotoxins are stored. those bufotoxins are released from the fat cells during that time, and they basically have a narcotic effect on the snake. Although it seems like when I handle hognose snakes, it's much more of a conscious decision for the snake to roll over and play dead than some uncontrollable interaction. So I'm not sure how much stock I place in that, which is why this says taxon may sequester toxins. So toxic organisms potentially get their toxins from their prey or provide their toxins to their predators, which might confer some protection to predators as they are being eaten themselves. When we think about a postmatism, remember that postmatism is this bright warning coloration. Because of that, mimicry can happen. So here you have a Pseudotriton rubor. What the hell is the common name of it? It's one of the spring salamanders. It's a plethodonid salamander. This is toxic. It creates toxic skin secretions. This is an eastern red-spotted newt, Notothalamus viridescens. It produces tetrodotoxin. Both of these species have overlapping ranges with the redback salamander or the red salamander, Plethodon cinereus. And so here you see a Plethodon cinereus that is presumably a mimic of at least two species that it shares a geographic range with. So we have a posematically colored species, and then we have a species mimicking it. Harvey Poe, one of the authors of your textbook, actually did this study with coral snakes and milk snakes throughout the tropics, and found that at any particular spot where you might find a poisonous coral snake, you can also find a corresponding non-poisonous king snake or other species that closely resembles it. So in both of these cases, the one on the left-hand side is a coral snake. You can tell because it has a more rounded head and there's less of a pronounced distinction between the head and the neck, whereas non-coral snakes tend to have a more pronounced head with more of a constriction at the neck. And so in all these cases, you have a coral snake on the left, harmless snake on the right, coral snake, harmless, coral snake, harmless, representing this mimicry complex within, in this case, Central America. I showed you this book earlier in the semester. There's a 2-volume set of books that I have in my office called The Venomous Reptiles of the Western Hemisphere. And in this book, they talk about coral snakes. And so here are a variety of coral snakes from around the New World. We see coral snakes that look like this and coral snakes that look like, well, I don't see our more local one. Anyway, this is one that occurs in the desert Southwest. And if you've ever heard the phrase red on yellow, kill a fellow, red on black, friend of Jack, that saying comes from coral snakes here. Red on yellow, kill a fellow. This is a coral snake. Unfortunately, that rhyme only works in the United States, because if you go down into the New World tropics, you get coral snakes that are black and white banded. You get coral snakes that are basically just red and black banded, another black and white banded one here. And at any given point in the New World, you might see a variety of these things. This basically looks like a California king snake in some ways. At any given point in the New World, while you find coral snakes that are patterned like this, you will find king snakes and other non-venomous snakes that look like this. And in some cases, they are red and yellow banded. Oh, sorry, that's Macrurus. So sorry, on the left-hand side, these are all Macrurus. On the right-hand side are harmless snakes that resemble coral snakes. So this complex represents a series of models and mimics, which brings us to the next thing that I want to make sure I don't forget. In any mimicry system, you have a model and a mimic. And in all cases, the model is the one that is either poisonous, venomous, or in some way dangerous. But in the case of the mimics can either be harmless or they can also be dangerous. What's the type of mimicry we have when we have a harmless mimic mimicking a harmful model? Harmless mimic mimicking a dangerous model. What kind of mimicry is that?
Is
Give
it
two
called
choices. Yeah, go ahead.
Batesian?
Yeah, it's called Batesian, named after Bates, who will describe this first. And then what is it called when a mimic mimics another dangerous species? It itself is dangerous, but it gains mimetic advantage by resembling other dangerous species in its area. It's also named after the biologist who described it. Anybody remember? Probably haven't heard this term since your first year.
Is it Malarian mimicry?
Yep.
Or did I just make that up?
Nope. Eulerian mimicry. So Batesian mimicry is where a harmless mimic mimics a dangerous animal. And Mullerian mimicry is when a dangerous animal mimics another dangerous animal. The thing about Mullerian mimicry is that if you're relatively rare, it benefits you to have more copies of organisms that look like you. And so over time, evolution will craft a series of dangerous animals that all live in the same geographic area that all resemble one another because they all get some advantage of there just being more of the model out there. In all cases, you generally need more of the model than of the mimic, because if you get too many members of the mimic species out there, the predators that are feeding on them don't associate that color pattern with being dangerous, because there are way more mimics than the models. So in all these cases, the models themselves need to be more numerous than the mimics. And Kiara and Rose will be familiar with this. Can you guys describe what's going on in this figure? This is once again a figure from your textbook, but they'll recognize this from their work.
Postmatism.
And crypsis.
Yeah, so this is Brody's work in Costa Rica. What is he doing here? Similar to something you've done.
Making clay snakes to test predation rates.
Yeah, so Brody tested this idea of whether or not these color patterns were actually aposematic by basically making clay models of different types of snake patterns and testing them versus a control, which is just a slate gray snake. And because he's making these things out of modeling clay, when a bird attacks the model, they leave indentations of the attack, so that you can lay these on the forest floor and you can say, well, this one is kind of cryptic, this one stands out. Well, if it stands out, it might be attacked more because you can see it more easily. But as it turns out, these models that are the color pattern of the coral snake got attacked considerably less often than the non-dangerous models. And there was actually a continuum of different types of models. And Kara and Rose did this with caterpillars, looking at plain green caterpillars that are a palatable species of caterpillar versus a banded pattern caterpillar like the monarch that is unpalatable. And they found a similar thing with caterpillar patterns. The monarch pattern was attacked much less frequently than the non-toxic type of caterpillar. So there is some advantage to being a particular color pattern. You just avoid predation because you have advertised the fact that you are dangerous. All right, that is what I have for you today. Tomorrow we will talk about conservation, but we are going to concentrate on conservation along a couple of lines of thinking. One line of thinking is presence of diseases. Another line of thinking is effects of climate change. And the third line of thinking is introduce species that may, for example, change the predatory landscape, introducing predators into environments where predators didn't occur. And then those predators either compete with the native species for food or actually feed on the native species and are driving species to extinction through direct predation. And that will set us up for Friday when we will be talking about the Miller paper. I am working on your last literature summaries so that I can hopefully have them out to you later on today or sometime tomorrow, so that you have feedback on those as you prepare your literature summary for Friday. Rose also reminded me that I need to get you out the format for how I want to get your collections and get all that locality data out to you as well from the places that we visited. So that's also on my list of things to do. I just haven't gotten around to that yet. So, any questions? All right, then go and stress about your final research presentation. And I don't think I'll see any of you tonight, because I think I'm in a different room. So I'll see you guys on Wednesday. All right, good luck tonight.